So now we are done with hypersensitivity. Let's move on to tolerance. So normally in the body, we don't have our own host tissue being attacked, right? So antibodies are produced, our host tissues are there, but these antibodies don't act against our self antigens. Why? Because there is a term called as tolerance. Our body is tolerant to self antigens. So this failure of tolerance is the one which is going to lead to autoimmune disorders. So self antigens are going to be destroyed in this autoimmune diseases because tolerance is lost. We have two types of tolerance, central and peripheral. Central is going to be present at the production side, let us the bone marrow and thymus. So in bone marrow and thymus, we have the central tolerance, and in the peripheral organs, we are going to have the peripheral tolerance. So in central tolerance, we have two mechanisms, negative selection or clonal deletion and receptor editing. So negative selection is going to be for T cells and receptor editing is going to be present for B cells. So negative selection is going to happen in thymus. So in thymus, we have a gene called as AIR gene. AIR stands for autoimmune regulatory gene. So this AIR gene is going to express some self antigens and it is going to test the T cells present in the thymus. So whatever T cells were actually acting against this self-reactive antigens they are going to be killed by apoptosis. Okay, so that they don't enter into the peripheral circulation. So this AIR gene is responsible for this negative selection or clonal deletion. Okay, so as the name suggests, negatively selected those T cells which were acting against the self antigens are going to be deleted. So that is why it is called as negative selection. So whenever there is going to be loss of function of this AIR gene, it is going to result in various polyendocrinopathies. It's called as a syndrome called as autoimmune polyendocrinopathy syndrome. Okay. In receptor editing, so again here the B cells are exposed to certain self antigens, and whomsoever is actually acting against the self antigens, they will edit their receptors. They will edit their receptors and change its configuration so that they don't react with self antigens anymore. So this kind of thing is called as receptor editing, seen in B cell central tolerance. In peripheral tolerance, we have many mechanisms, starting with energy. Energy can either be of T cell energy or B cell energy. Energy is nothing but they don't react. They are just they don't react to anything. So normal cells, usually whenever a normal cell is present, for T cell activation, I already told, two stimulus is needed. One is the MHC interaction. Second one was the co-stimulatory signal. So whenever a normal cell is going to react, self antigen is going to come with the T cell, this co-stimulatory signal will not be present. Even though MHC signal can be present, the co-stimulatory signal, that is the CD8086 with 28 will not be happening. So without the co-stimulatory signal, T cells cannot be activated, resulting in T cell energy. Secondly, there is something called as co-inhibitory signal. So in T cells, we have certain receptors called as PD1 receptors and CTLA4 receptors. So the CTLA4 is actually similar to the CD28. Okay, so CTLA4 is like CD28 only. So the CTLA4 has much more potency than CD28 to bind with this CD80 or 86. Okay, so they are going to bind with the CD80-86, and when this interaction of CD80-86 with the CTLA4 happens, it is going to inhibit the T cell. Okay, it is a co-inhibitory signal. So this CTLA4 and 8086 is going to be an inhibitory signal. Okay, while the PD1, its ligand is going to be PDL1, which is programmed death ligand 1. So PDL1 ligand is going to attach with this PD1 present on T cells, and that again will result in the suppression of the T cells. So both CTLA4 and PD1 are co-suppressors. Okay, so they are going to actually, in their interaction with their corresponding ligands, will actually cause the suppression of the T cells. It is very important in neoplasia chapter as well. So that time we'll be learning about this more in detail. And then the third mechanism is going to be the T regulatory. So before that, I'll mention about the B cell energy also. So B cell activation was because of the CD40 and CD40 ligand interaction, right? So this interaction cannot take place when a normal cell is going to be presented. So without this, the B cell interaction, B cell activation cannot take place. So that will result in B cell energy. So the second mechanism of energy is T regulatory cells. So as I mentioned, these are the major regulatory cells responsible for controlling the immune system. So as I mentioned, these Treg cells are going to be positive for CD4 because they are a subset of CD4 positive T helper cells only. And apart from that, they are going to be positive for CD25 and FOXP3, right? Remember that. So whenever there is going to be a polymorphism of this CD25, it is going to result in certain autoimmune diseases like multiple sclerosis. So in multiple sclerosis, we have the CD25 polymorphism. And in FOXP3, so mutation of this FOXP3 is going to result in a syndrome called as IPEX syndrome. So IPEX is the short form actually. It stands for immune dysregulation, polyendocrinopathy, enteropathy associated X-linked disorder. So it is an X-linked disorder in which you have immune dysregulation, polyendocrinopathy and enteropathy. Okay, so FOXP3 mutation results in IPEX disorder. So normally also we have commensal bacteria in our body, right? So against this commensal bacteria, we are not mounting a immune response. So how does it happen? Because Tregs are controlling the immune response. Again, same thing for a fetus also. Fetus, even though it is a foreigner, alien to the body, but still it is not being killed because this is going to be controlled by the Tregs. Okay. So the third mechanism of tolerance is going to be apoptosis. So whenever self-reactive T cells are being exposed to the self-antigens, they are going to be killed either by fast acceptor, which is the death receptor pathway, or they'll express BIM and then killed by the mitochondrial pathway. So self-reactive T cells will always be killed by this apoptotic pathway only. So mutations in this fast expression is going to be resulting in a syndrome called as ALPS. ALPS stands for autoimmune lymphoproliferative syndrome. Okay, so we have three syndromes already. AIR gene, which was central tolerance, right? So AIR gene mutation will result in autoimmune polyendocrinopathy. FOXP3, which is Tregs related mutation, is going to result in the IPEX syndrome. Then we had ALPS. Here ALPS is because of mutation in the FAST ligand mutation. Then antigen sequestration. This is another type of tolerance only. So in the body, there are some sites called as immune privileged sites. Basically, there are these antigens are not exposed to the circulating blood. Okay. So where are these immune privileged sites? They are going to be present in the brain, eye, testis. So remember it as BET, brain, eye, testis. So these antigens are not usually exposed to the circulating the cells. Okay, so circulating blood they are not exposed to. But whenever there is a trauma to any of these organs, the antigens are going to be exposed to the circulating blood. So this is going to cause a very high immune response. And that is why if there is trauma to the eye, so the post-traumatic uveitis, even though only one eye is affected, one eye is having a trauma, the other eye is also going to have a reaction because the antigens are being exposed to the circulatory blood now. Okay, so that is why orchitis and post-traumatic uveitis, even though the trauma is unilateral, it is going to cause a bilateral reaction.